Welcome to All-in-One Pharma. Today we are going to discuss about the preclinical studies. In the last class we studied about the new drug development. In that we have the few steps. First one is the synthesis and isolation. Second step is in preclinical studies. Next after clinical, then marketing. We are going to study about the preclinical studies in detail. In the last class, I already said about the preclinical studies, which means preclinical studies, the drug which are testing on animals is preclinical studies. We are testing the drug on animals using some of the tests given below. This is about the preclinical studies. We studied in detail about the preclinical studies. First, mainly, what is preclinical studies? Preclinical studies means after synthesis and identification of a prospective compound, series of compounds is tested on an animal to expose the whole pharmacological profile. I already said about this, which means in a new drug development we have some steps, right? In the first step was synthesis and identification. After synthesis and identification of a prospective compound series, we have to test the compound on animals to know about the pharmacological profile of the drug. We have two kinds of pharmacological profile: pharmacodynamics and pharmacokinetics, right? First, drug reaches to our body. It undergoes the absorption, distribution, metabolism, and excretion. This is about the pharmacokinetics, and we have to know about the drug before itself, that what drug to do towards our body. We have to test it in some of the animals, right? Without testing on animals, we can't give it directly to the humans because it gives some side effect to us or side effect to us. Because of this reason, we have to test the drug to the animals, and we have to undergo the preclinical study. This is very important in the new drug development, is preclinical studies, which means I already said that the drug have to be tested on the animals of the pharmacological action. Okay, next, experiments are generally performed on a rodent and large animals. I already said that we have to test on the animals, but what type of animals we have to test, we have to choose that also. First, rodent type of animals like mouse, rat, guinea pig, and rabbit. These are the four animals which are widely used for the testing of the drug, which have permission. We have permission from the new drug development and the government. And larger animals like cat, dog, and monkeys. This is more larger animals, but mostly we can perform this type of experiments on mouse, rat, guinea pig, and the rabbit. This is about the preclinical studies. This is a general introduction about the what we can do in these preclinical studies. Next, what are the tests we have to do in these preclinical studies? First, we have following steps. First step is screening test. Next, the testing on isolated organs by bacteria cultures, and third one is test on the animal models of a human disease. Next, we have few of the tests like confirmatory test, mechanism of action, qualitative test, pharmacokinetics test, toxicity. To know this all actions, we have to do the drug test on an animals. First, about the screening test. First, mainly we can do this is a screening test. What is about this screening test? In this test, they are the simple and rapid performed test to indicate the presence or absence of a particular pharmacodynamic activity that is sought. In this screening test, we came to know about the pharmacodynamic activity. We can give the drug to the animals. We have to test the pharmacodynamic activity in salt. Example like analgesic, sedative actions. If any drug we given to that animal, if that animals have slowed their activity, which means it is undergoing the analgesic activity, right? Like this, we have to find out the screening testing. Next was test on the isolated organs and bacteria cultures, etc. Means we have to detect the specific activities like antihistamine, antisecretory, vasodilator. This type of activity we have to test on the drug which have type of activity like antihistaminic or antisecretory or vasodilator. We have to test in the bacteria cultures by using the bacteria culture. This is on the test on the isolated organs and bacteria. First one screening test, second one is isolated organs, bacteria cultures, etc. Next one, test on the animal models of a human disease, which means we have to choose the animal which have the human disease. example, take a mouse is suffering from the tuberculosis. We have to give the drug to cure the tuberculosis, right? We have to take the animal which having the human disease. This is about the third test. We have to test on the animals having the human disease. We have to give the drug, and what is the action is undergoing the animal. This is about the third test. Next, we have few of the tests like for confirmatory test, which means the drug having which type of confirmatory, which is active compound or passive compound, destro compound or levo compound. We have to confirm the confirmatory test. Next, the mechanism of action. When the drug have each drug have the specific mechanism of action, right? We have to find the mechanism of action of specific drug by testing to the animals. Next, the qualitative test. The drug have some quantitative, which means dose. We have to know about the dose also, right? If we give the high dose to that animal, the animal will get some of the side effect. We have to know the optimum dose. By testing on the animals, we can know the optimum dose. This is about the quantitative test. Next one is pharmacokinetics. I already said that the pharmacokinetics is very important, what drug does to our body, which means ADME parameter, absorption, distribution, elimination, excretion. That 4 parameters we can understand in the pharmacokinetics. Next one is toxicity. Toxicity means every drug has the pharmacovigilance activity, right? Which means the adverse drug reaction. Some of the drugs have side effects also. We have to, some of the drugs give the toxicity to of the body. We have to know the drug is really giving the toxicity or not. We can find this type of things by using the preclinical studies. When we are doing the preclinical studies only, we came to know the confirmatory test, mechanism of action of drug, and the quantitative test, and the pharmacokinetics activity, and the toxicity. By knowing this type of side effects or any problem having the drug, we can know and we can correct it by some of the implementations, because we have to do the preclinical studies. This is about the preclinical studies, and thank you. Please subscribe to our channel.